respiration. And I'm telling you now, cellular respiration and all the details of it is probably the hardest thing that we learn in AP Bio. Okay, it's just there's a lot of details and there's a lot of parts and there's a lot of steps. So make sure you spend the time with it. It's huge. Okay, so we're just gonna dive right in. So cellular respiration, every living thing on planet Earth does this. Okay, so plants, animals, everybody. And what they do is they're gonna take glucose made by photosynthesis, and and some oxygen, and they're going to turn it into some carbon dioxide, which they don't really want, usually a waste product, and a little bit of water, great. And here's the key part, energy. And we know that energy in the cell is in the form of ATP, okay? So that's kind of the whole deal. Now there's a lot of this thing of, in this process, there's a lot of this oxidation and reduction. So let's just real quick review. Oxidation means that something loses electrons. And reduction means that you gain electrons. You get a reduction in charge, okay? So in every reaction, there's something that gets oxidized and there's something that gets reduced. So in this case, the X is getting oxidized because it's losing its electron, so it's oxidation. And the Y molecule is getting reduced because it's gaining its electron, okay? Reduction. So when you look at different formulas, when you can use methane as a source of energy, something is getting oxidized. That's going to actually be our carbon. And the oxygen is going to be reduced. Okay. Same thing is going to happen in cellular respiration. The carbon is going to be oxidized, just like before. So I have some oxidation occur. It's going to lose electrons. And we're going to see that the oxygen is going to be the final electron acceptor and gain electrons and turn them into water. Okay? So that's kind of the oxidation reduction part of this whole thing. Now, we're going to meet another guy, another molecule, and you guys have already met a very similar molecule, and it's NAD+. We met NADP+. In photosynthesis, very similar. This is an electron carrier, okay? I always remember it is the NADP is in photosynthesis, and photosynthesis starts with a P. So it's an electron carrier. So he's going to carry around that electron, okay? And it acts as a coenzyme, just like NADPH acts like a coenzyme, okay? So that's kind of the deal with this guy. So we're going to see him carrying around electrons a lot. Now, we're going to use these two terms, and we're going to see this two-term thing happen, where we have these two things called phosphorylation, and we've actually seen one of them. One is called substrate-level phosphorylation, and in this one, ATP is going to be produced. Actually, in both of phosphorylations, ATP is going to be produced, okay? And what happens in this one is we have a direct transfer of that phosphate, with the little inorganic phosphate, or straight phosphate, from an intermediate compound, or just from a compound, to ADP. Okay? The other type of phosphorylation that you might see is called oxidative phosphorylation. In oxidative phosphorylation, we're going to have the same thing. ATP is going to be produced. And what happens in this one is we have an exergonic, that releasing of energy, slide, we call it an exergonic slide, of electrons is used to create a hydrogen gradient. This sounds somewhat familiar. We might have seen this before. Okay? And we use the energy, use the kinetic energy more specifically, of hydrogens going down their concentration gradient to create ATP. We've seen this before. We saw this in the light reaction of photosynthesis, and we're going to see it again today in a section called oxidative phosphorylation. Okay. Before we dive into all the parts, we're going to look at the different parts of the mitochondria, and we're going to look at the structure of a mitochondria. So we're going to attempt to draw a mitochondria here. So mitochondria, you should be are a big, large organelle, membrane-bound organelle. And obviously, if you're a prokaryote, you're still going to do cellular respiration. You'll just do it on the cell membrane, just like a photosynthetic prokaryote would do photosynthesis on the cell membrane. And it has this really lots of infolding, and I barely drew any of the infolding, but there's tons of infolding to increase the surface area, okay? And so this is really tricky naming here. This is called the outer membrane, and I bet you can guess what this is called. This is called the inner membrane. Oh, they've got real creative here. The inside, this part here is called the matrix, okay? And then this space in between the outer and the inner membrane is called the intermembrane space. I'm telling you, they got real creative. And the only other thing that I want to point out is if you were to look at just one of these little folds, and there would be thousands or even millions in one mitochondria, one of those little folds is called a cristae. And the only other thing I want to show right in here is just where things happen, okay? Outside of the mitochondria is actually where step 1 happens, which is glycolysis. And then step 2 is going to happen here, which is the Krebs cycle. It's also known as the citric acid cycle. And then inside the membrane, so I'm going to kind of draw a little arrow, is step 3, which is the electron transport chain. Inside the inner membrane, I should say. So there's kind of 3 major steps. There's one that, we'll talk about this, but there's one that happens between step 1 and step 2, okay? So let's dive in and start to look at these steps. So this first page is the first step, which is glycolysis. And remember, glycolysis occurs in the cytosol. So this is occurring in the space of the cell, okay? There's kind of 2 main phases. We're going to invest some energy, and then we're going to lose some energy. The first thing is we have this right here is glucose. Okay, so here comes glucose. It enters in. And we're going to use some ATP. I'm going to do ATP in red today. And it's going to turn into ADP. We actually have to invest some energy in. So we're going to follow that little phosphate. So the rest are carbons. That's a little phosphate. So then we're going to rearrange it a little times. And when we rearrange it, we're using enzymes to do that, of course. And then, we're going to have another ATP come in and drop off another phosphate. So now we have 2 phosphates on here, okay? And then we have another enzyme that comes in and splits it. Guess what? You don't need to know the names. And so now the exact same process is happening on either side. And so we have a 3-carbon sugar with a phosphate attached. And at this point, we're actually going to add another phosphate. Is going to come in, another phosphate. And our friend, do it in blue, NAD+, our new friend, NAD. And it turns into NADH. Okay, so it's going to pick up an electron, and it turns into NADH when it did that. Okay, so now at this point, I've added this phosphate, so now I have 2 phosphates. Yay, 2 phosphates. And that's not a phosphate from ATP, it's just a phosphate. Okay, at this point, I'm actually going to make some ATPs. This is that substrate level phosphorylation, because I'm doing it as a direct transfer from a compound. Okay, now my molecules only have 1 phosphate on them, and we kind of do some rearranging by some enzymes that we don't have to know the name of. And finally, I make some more ATP. And I end up with these 2 molecules down here, which are called pyruvates. Okay. So, on the next page, we're going to just summarize everything that just happened. Basically, I made some ATPs, and I rearranged some things. Okay, split glucose, all that kind of stuff. So, the first stage, up at the top before I split everything, is what we call the energy investment stage. And it uses 2 ATP molecules. And I'm going to split glucose. Glucose is split. Okay? And in the energy yielding phase, I'm going to produce, that's the second part, I'm going to produce 4 ATP molecules, but I used 2, so I'm only gaining a total of 2. And I'm going to write down, just to clarify, how did I make those? I made them by substrate level phosphorylation. So just to summarize, in went glucose and 2 ATP, and out went 2 pyruvates, 4 ATP, and 2 NADHs. Okay, but I had to put 2 ATP in, so I'm only going to net 2 ATP. Okay, so now we're going to follow these pyruvates. We're going to deal with these guys later.